Ngalakin Khadijan Nyunga Boja.
Hello everyone, my name is Martha Ludwig and you might remember me from previous lectures. I'm going to be taking the next 6 lectures of BIOC3005, and in those lectures we'll talk about energy generation and energy storage. But before we start talking about today's lecture material, I'd like to just remind you about this Have Your Say survey, and you should have all got an email, so check your inbox for the link to the survey, because we'd really like to get your opinions, your ideas about how we're going at UWA, how you're going with tertiary education. So for today's lecture, as I said, we're going to focus on energy generation and storage, and where we're going to start is talking about mitochondria. So we'll talk about mitochondria, the structure in today's lecture, also electron transport. Now, some of you might remember that we talked about this at level 2, so if you were in BIOC3002 last year, you might remember some of this information. So this is kind of presumed knowledge, and so we'll go through some of the slides relatively quickly because, again, you saw it last year. If you need to contact me, then here are my contact details. So I'm part of the School of Molecular Sciences, and you can find me in either 2 different places. So you can find me in my research office, which is on level 3 of the Bayless building, and the extension is there if you need to ring me. Or you can find me in the head of school office, which is on the ground floor of the Bayless building, and the extension, the direct extension, is there for that room. I am happy to meet with you if you have any questions about any of the material that I present, so please don't hesitate to email me. I will find a time. I don't have regular office hours, but I will certainly find a time when we can get together and we can sort out any questions that you have. So when I'm not teaching, when I'm not being head of school, I do spend my time in research, and the research focus of my group is the molecular evolution of photosynthetic systems. And so we use the recombinant DNA technology to basically discover genes, to find out the protein function of what those genes code for. We use PCR, we use quantitative PCR, DNA sequencing. In fact, we're now getting whole genome sequencing of the plants that we work on, which happen to be Australian native plants that are very interesting because some closely related members of this particular group do one type of photosynthesis, another does a more evolutionary derived type of photosynthesis, and then there's one species that does a type of photosynthesis that's in the middle. So we can actually see changes at the level of the gene and the proteins that led to the more ancestral state to the more evolutionarily derived state. And that evolutionary derived state allows plants to survive where it's hot, dry, there's high light environments. So we use immunocytochemistry to pinpoint the localization of those proteins in a cell, in the tissue that we're studying, and then we generate antibodies to be able to do that as well. And we look at the light microscope level, we look at the electron microscope level. If you're interested in any of those techniques, technologies, if you're interested in the project, please don't hesitate to see me. So I've put some suggested background reading on the next slide, and really this is reading out of the textbook. So this is what I think is background knowledge that you should have to be able to understand the material. And it's presented really well in Leninger and then also in Buchanan. I'll talk about the specific differences that the mitochondria of plants have compared to the mitochondria of animals and some other eukaryotes. And then what I'd like to do is basically use the next 2 lectures to give you the base of what I think are some interesting developments in the area of mitochondria and energy generation and storage. So for the live workshops, I've put a review paper here on the slide that I'd like you to read and then I'll have some questions which I'll put on the LMS, and I'll also put up this notice on the LMS as well, so that you can just go directly there and have an announcement of what we're going to do. But at the workshop, there'll be those questions that you'll know ahead of time, and I'd like to, if possible, form groups at the workshop so that then those questions can get discussed, and then the answers can be presented to the whole group. All right, so have a look at all of the questions, be able to answer all of them, but then keep in your mind that you're going to be discussing them with your cohort, with the other people in the class, and presenting that information, right? The information that we talk about, so the questions, that is material that is accessible, okay? So don't think that this is just some, extra thing that I'd like you to do. This is part of understanding mitochondrial function and energy generation and storage. I've also listed here on this slide the learning outcomes for this lecture, so you should be able to answer these questions after the lecture, and any exam question that I ask or develop will certainly focus on these learning outcomes. Okay, so on to the lecture material. The structure of mitochondria. You would have seen this in a number of units by now. You know that mitochondria are surrounded by two membranes. There's the outer membrane that has about 30 to 40% lipid in it, 60 to 70% protein, and importantly, on that outer membrane there are structures known as porins. And again, you might remember from an earlier lecture, I believe I mentioned it in Sci 1106 lectures, that porins have beta sheets. That's their secondary structure. And so basically they form a barrel structure in that outer membrane that's open on the two ends. So there's no top or bottom to the barrel. And that allows allow molecules that are less than about 10 kilodaltons to get through the outer membrane. But then that inner membrane, okay, that inner membrane that encloses the matrix, the interior of the mitochondria, that membrane is impermeable to molecules and ions. So there's lots and lots of transporters in the inner mitochondrial membrane. That means that the inner membrane of the mitochondria has a lot more protein in it. So it's about 80% protein, okay, because of all these transport molecules. And also because that's where the respiratory chain proteins are. So the inner membrane of the mitochondria is just packed with different types of proteins. Of course, the ATP synthase is also on that inner membrane. The inner membrane, as shown in the diagram, infolds into the cristae, so those tubular structures, sometimes also sheet-like structures, and then those cristae project into the matrix, as I've said, that inner part of the mitochondria. And in the matrix, that's where the enzymes of the TCA cycle, remember that's also known as the Krebs cycle or the citric acid cycle, that's where those proteins are, that's where that cycle activity takes place. But fatty acid oxidation also takes place in the matrix of the mitochondria. And remember, mitochondria have their own DNA, and so mitochondrial DNA is in the matrix. All the machinery to transcribe that mitochondrial DNA and then to translate the messenger RNAs that are transcribed from the DNA are there. So the basic genetic mechanisms for mitochondrial DNA and protein synthesis are in the matrix as well. Now, I think the technology might fail me on the next slide, so I'm going to see if I can change to the if I can change to this full screen for a minute so I can do the animations here. So the structure of mitochondria as we've seen in that previous diagram, has the outer membrane and the inner membrane, and the inner membrane folds into those Christie membranes. So the inner membrane and the Christie membranes are continuous, right? And the early structural images from the electron microscope showed that there were quite wide openings where the infolding of the Christie membranes and the outer membrane occur. Okay, so a big space, broad openings from the Christie to the inner intermembrane space. We now know that this really isn't what's happening in the mitochondria. Okay, that's not the true structure. That in many cases what we were looking at were artifacts. And what we know now is from a technique called tomography, and that's using, again, the electron microscope, but it looks at the preparations for the electron microscope in sections that are then reconstructed, so thinly sliced sections of a mitochondrion, in this case, that are then reconstructed to give a three-dimensional image. And what we found is that the inner mitochondrial membrane actually has two very distinct subdomains. The inner boundary membrane, also known as just the boundary membrane in some articles, some textbooks, and that inner boundary membrane interacts with the outer membrane, and it has to do this in a coordinated manner when mitochondrial division occurs. And also, you might remember that mitochondria can fuse to one another. So they can divide, they can come back together and become a larger mitochondrion. So we can see in the electron tomographs the outer membrane and the inner membrane, and we can see that there are what appear to be contact sites, but again, people are arguing that these are artifacts. Okay, so just keep that in mind. And then there's the Christie membrane. So that's the second subdomain of the inner mitochondrial membrane. So the Christie membranes, as we've known from early electron micrographs, do form from invaginations of the inner membrane. So there's invaginations of the inner boundary membrane, and these Christie can form tubules or sheets, which we recognized in early electron micrographs. They project into the matrix, and as it turns out, these Christie membranes make up most of the inner membrane surface. Okay? So if you go to the tomograph level, okay, and look at these reconstructed mitochondria, then you can get images like these pseudo-colored images shown on this slide. And you can easily see how individual Christie can form tubules or sheets, as shown in image B in the top left diagram. Okay, so sheets of mitochondria, Christie. And you can see the outer membrane shown in purple, the inner membrane shown in light blue, and also those where the Christie join the inner membrane, and those are the Christie junctions. They're narrow tube-like connections with the inner boundary membrane. And notice again in panel B on the top left, and also on the lower right-hand side, the figure A, that those Christie junctions can be multiple, or there might just be one. And so we can see the Christie coming off, for example, and the lower figure A, you can see the single Christie junction at about 3 o'clock leading into this kind of tubule, but then that goes into more of a sheet like Christie. But then if you go to about 12 o'clock and 1 o'clock on that same diagram, you can see the same Christie having two connections with the inner boundary membrane. Right? Other information that you can get from these tomographs is shown on the right-hand panel A, so upper right-hand part of the slide. And you can see the connections that are the red circles. Those are each of the Christie junctions. So the outer and inner membrane has been stripped away from this mitochondrion image, and yet we can still see those Christie junctions where the Christie would join to the inner membrane of the mitochondrion. So if you have a chance, have a look at the tomograph video that is on YouTube, and you can see the three-dimensional reconstruction of the mitochondrion through electron tomography. And I think it's just a different way of thinking about mitochondria and how they're put together and how they work. And I think it will help you understand the review paper that I'd like you to read for the live workshop, because there's a definite connection between structure and function. And of course, that's what we like to study in biochemistry, structure and function. And we'll come back to a bit more about the structure-function relationship later in this lecture. And as I've said, it's certainly a big part of the article that I've assigned for the live workshop. All right, so getting on to the function of mitochondria. I think every textbook, every article about mitochondria says something like they are the powerhouse of the cell. Yes, they are the compartment that specializes in ATP synthesis. Okay? They are the sites of what's known as aerobic respiration. Nearly all eukaryotes have mitochondria, not all. Nearly all eukaryotes have mitochondria. Aerobic respiration, the definition of that process, is the controlled oxidation of reduced substrates to carbon dioxide and water. And in the case of mitochondria, this is linked to ATP production. And so that whole process, okay, is known as oxidative phosphorylation. And as I've said, most eukaryotes are able to carry out this process. Now, there's a couple of nuances, okay, a couple of subtleties that I'd like you to realize. That if we're talking about ATP production in the cytoplasm through glycolysis and the production of GTP, which can be considered an ATP equivalent, in the mitochondrial matrix, then we're talking about substrate level phosphorylation. Okay, that's different than oxidative phosphorylation. Oxidative phosphorylation happens through electron transport coupled to ATP synthesis. Okay, so in animals, we see the respiratory substrates being proteins, fats, and carbohydrates. Again, as shown in the diagram on the slide. So at the top, you can see the proteins, polysaccharides, fats, all leading into the cell. So that's shown in gray. So we see the cytosol in gray. And that's where glycolysis happens. And then pyruvate, an outcome of glycolysis, can be transported into the mitochondria. And there in the mitochondria, it can enter the TCA cycle. So here we're talking so far just about substrate level phosphorylation. And then the outputs of the TCA cycle and glycolysis can be used in oxidative phosphorylation to generate water and also ATP. Okay. In the case of plants, then we're talking about the respiratory substrates being sucrose and also starch, which of course is glucose molecules linked together. Now, when we look at that breakdown, okay, of glucose. We also have to remember that not all glucose molecules entering glycolysis will be completely oxidized. And that's because some of the intermediates that are produced in glycolysis and also the TCA cycle are used to make other compounds. They're used to make amino acids, nucleic acids, fatty acids as well. And so the diagram on this slide shows those different branch points and how different amino acids are made, and how fatty acids and some nucleic acids, they're also included in the diagram, right? But for the glucose molecules that are completely oxidized, the energy that is produced is the driving force for ATP synthesis, okay? And so we see more than 5,500 kilojoules for every mole of sucrose being oxidized, being produced when it is completely oxidized. So a strong thermodynamic driving force for the production of ATP synthesis. A bit more about that and the linking of these different processes, the linking of substrate phosphorylation with oxidative phosphorylation is shown in this slide. So we've got, again, a diagram on the right-hand side of the slide showing the breakdown of glucose in the cytosol of the cell, and then NAD being produced, as well as pyruvate. As I said, pyruvate is transported into the mitochondria where it can enter the TCA cycle, and then the products of the TCA cycle, ATP or GTP equivalents, and NADH, FADH2, and the NAD that's produced through glycolysis in the cytoplasm can then enter oxidative phosphorylation, can enter the electron transport chain of the mitochondria, and that can lead to the production of the proton motive force, which drives the production of ATP synthesis. Okay? So electron transfers on the mitochondria, electron transport chain is linked with the translocation of protons across the inner mitochondrial membrane at the cristae, as shown in the cartoon, and then that leads to the generation of ATP through the ATP synthase. A nice way to remember the direction of electron flow is shown in that left-hand diagram on the slide, and basically it shows that all molecules that are involved in electron transport are between the substrate, NADH, and the final acceptor, which is oxygen to make water, with respect to the reduction potential. Okay? So we can think about moving down a redox potential scale from a negative value, where NADH is oxidized to NAD, to a more positive value where oxygen is reduced to water. Okay, so that might help you remember the direction of the flow of electrons through the electron transport chain. And the direction is one way, okay, going from that negative redox potential to the positive redox potential. A reminder about what makes up the mitochondrial electron transport chain. There are 4 large multi-subunit complexes, and the simple names you might remember are complex 1, 2, 3, and 4. Okay, so those are the simple names. If you want to remember what those complexes actually do, then take the time to learn the full names. So complex 1 is the NADH dehydrogenase, complex 2 succinate dehydrogenase, complex 3 cytochrome BC oxidase complex, or cytochrome BC complex, and then complex 4 the cytochrome C oxidase. Okay? And remember, there are also 2 mobile electron carriers, and so those are ubiquinone, that's the oxidized form, ubiquinol, the reduced form. That's also known in some textbooks and some journal articles as coenzyme Q, or simply Q in some diagrams. And then the second mobile electron carrier is cytochrome C, right? So all of those 4 complexes are shown in the diagram on the slide. And this is a very linear model of the mitochondrial electron transport chain. This is how it is depicted, I think, in every textbook and in many journal articles. But although it's easy to remember in this way, it's not really what's happening inside mitochondria. And that, again, is illustrated in the review article that I'd like you to read for the live workshop. Okay? Again, it should allow you to realize the structure-function relationship of mitochondria and electron transport and ATP synthesis, but also move you away from this, two-dimensional images that we see in textbooks that are easy to explain, but not really how things function in the real world, right? Now notice also that in this diagram, there's the ATP synthase. That's also called complex 5. It's not really part of the electron transport chain because it doesn't have anything to do with electrons. It transports protons, okay? But very often people will call it part of the electron transport chain. It's part of oxidative phosphorylation. That is abbreviated as OXPHOS. That is true. Okay, ATP synthase is part of oxidative phosphorylation, or OXPHOS. Okay, so another review slide is up next. And this is just to remind you that the electron carriers in the electron transport chain are attached to proteins, and they are known as prosthetic groups or centers. And that's what's actually accepting the electrons, donating the electrons to the next part of the electron transport chain. And so we talk about flavoproteins. So those are proteins that have flavin nucleotides attached to them. And again, this is information that you would have seen in BIOC3005. So the flavin mononucleotide, FMN, the structure is on the right-hand side of the slide. It's got that single complex ring structure, so it's a mononucleotide. I will never ask you to draw these molecules, but as biochemists, if somebody says a mononucleotide, this is the kind of image that should come into your head. If people talk about a dinucleotide, which is in fact what FAD is, okay, then there's a second complex ring structure, and that's shown again in the diagram on the right-hand side of the slide. And remember, FMN and FAD are able to carry, be involved, transfer, whichever word you like to say, 1 or 2 electrons, which means that they have intermediate structures. Okay, they can exist in an intermediate state where they've accepted 1 electron, and then they can be in a fully reduced state where they've accepted 2 electrons. Cytochromes, you might remember, have that heme structure, so a ring as the prosthetic group, again attached to proteins, but it's the prosthetic groups that's involved in electron transfers, and single electrons are transferred through these prosthetic groups, or the heme structures. And there's different types of hemes, so there's different types of cytochromes: cytochromes B, cytochrome C, cytochrome C1, okay, cytochrome B6, and so on. And then there is the iron sulfur proteins. So they're also involved in single electron transfers, and you can see that the iron group is coordinated by sulfur atoms, and they can be quite complex structures. But again, those groups are attached, associated with proteins, right? Okay, so that's review, and the next few slides are also review. So we've got here the structure of the first complex in the electron transport chain, and that is complex 1, so the NADH dehydrogenase. It contains a flavin nucleotide, so FMN. It also has some iron sulfur clusters, and in the diagram N2 is illustrated. That is an iron sulfur cluster. okay, just to be clear. And it's a large complex. And again, I think I made this point in BIOC3002. Lots of polypeptide chains in it, so a multi-subunited structure. And it's easy to recognize because it's got an L-shaped structure with one arm of the L being embedded in the membrane, the other arm sticking into the matrix of the mitochondrion. It's very large, so it's at least 650 kilodaltons. In plants, it's even larger than that. I don't expect you to remember that kind of detail. It's putting it in context, okay? Complex 1, the NADH dehydrogenase, is a big multi-subunit protein complex, okay? That's what this slide is all about. The second important part of this slide is where the electrons come from that are then transferred through the protein complex, okay? And they come from NADH. That's the electron donor, strong electron donor, and it gives up 2 electrons that then are transferred through those iron sulfur clusters from the FMN associated protein and ultimately to ubiquinone, okay? And so that's identified in the image as Q, okay? So that's the unreduced form, the oxidized form, and when it takes up the electrons, it becomes QH, or ubiquinol, and as I've said, it's QH2 in the diagram, right? A third important part of the NADH dehydrogenase is that it transfers 4 protons from the mitochondrial matrix to the intermembrane space, okay? And that's for every NADH that is oxidized. I've also made a note here that in Leninger, some editions of Leninger, the diagram is incorrect, and I pointed that out again in BIOC3002. You can have a look and see where it's incorrect, but remember, I'll give you a hint, it was incorrect on the NADH side of the transfer, okay? So what's shown in this slide is the correct direction. NADH is oxidized to NAD plus a proton. Okay, so that's complex 1. Complex 2 is succinate dehydrogenase. It again consists of a multi-subunit proteins. It has a molecule of FAD, so if we're talking about what's involved in the electron transfers, FAD, as well as some iron sulfur centers and also a heme group. That heme group actually plays a protective role in case there's too many electrons going through the electron transport chain. That heme B appears to grab onto some of those electrons to stop the electron transport chain from being over oxidized, basically, or over reduced, to balance oxidation and reduction reactions. Okay, so complex 2 is smaller than complex 1. It's made up of 4 subunits, and they're illustrated as A through D in the figure. So A and B extend into the matrix, and that's where the binding sites are for succinate, and also that's where the FAD molecule is, so that's the FAD that gets reduced to FADH2. And then the CD parts of the complex are what are embedded in the mitochondrial inner membrane. Remember, succinate dehydrogenase, complex 2, is also part of the TCA cycle. So it might sound familiar from another angle, and that is because it's part of the TCA cycle. So when succinate is oxidized to fumarate, then FAD is reduced to FADH2, and then that allows the electrons to move from FADH2, which again is a strong electron donor, through those iron sulfur complexes, and ultimately the electrons end up at ubiquinone. So they end up in the ubiquinol pool, or the UQ pool. You'll hear those different phrases and read those different phrases. Okay, so the electrons end up in the same place as where electrons end up from complex 1. Okay? But notice that in complex 2, no protons are translocated across the inner mitochondrial membrane. So from the UQ pool, electrons are then transferred to complex 3. And complex 3 is cytochrome BC complex. So that already tells you that there are lots of heme groups involved in electron transfers in this particular complex. There are also, however, some iron-sulfur centers. Okay? So the complex is made up of 2 identical monomers, so it's a dimer in a functional complex. Each of the monomers is 250 kilodaltons, so about the same size as complex 2. Again, smaller, even put together, even as a dimer, it's smaller than complex 1. So complex 1 is still the biggest, right? There are 11 different subunits in complex 3, but interestingly, the function of only several of them are known. Okay, the others, we just don't know what they do, right? So in complex 3, we see the transfer of electrons from UQ, and that transfer of electrons happens in the interior of the membrane, because remember, UQ and ubiquinol, the reduced form, function within the plane of the mitochondrial membrane. So here in the cavern, okay, so that's the part of the protein complex, so complex 3, where UQ is, okay, that's where the electrons are donated, are given up by ubiquinol, and the acceptor is the risky iron-sulfur compound or complex. So the risky iron-sulfur part of the complex 3 is mobile, so it can move into the plane of the mitochondrial membrane, take on those electrons from the UQ pool, and then transfer those electrons to cytochrome C, which is the second mobile carrier located on the intermembrane side of the mitochondrial inner membrane. Okay. As those electron transfers happen, we also see 4 protons moved from the mitochondrial matrix to the intermembrane space, and this is for every 2 molecules of cytochrome C that are reduced. Okay, so keep the stoichiometry in your mind. Okay, what's getting reduced? How many electrons are moving? How many protons are moving at these different complexes? All right, so now we've got the electrons on cytochrome C, which is that second mobile electron carrier in the intermembrane space, and cytochrome C then interacts with complex 4, the cytochrome C oxidase, on the intermembrane side of the complex. There are heme groups in complex 4 as well as copper ions that are involved in electron transfers, and they're illustrated in the diagram. Again, each of the monomers of complex 4, which again functions as a dimer, is about 200 kilodaltons, lots of different subunits, and again, we know what subunits 1 through 3 are, what their functions are, so they're necessary for electron flows, but the other 10 subunits, again, their role, what they're actually doing, it's not really clear. Potentially, they're involved in regulatory roles for electron transfers. Okay? So when electrons are transferred from cytochrome C through complex 4, they end up at oxygen, so the final electron acceptor is oxygen, reducing oxygen to water in the mitochondrial matrix. Okay? 4 protons are used in the mitochondrial matrix for the reduction of oxygen to 2 molecules of water. Okay? Again, keeping the stoichiometry in mind. But then also notice that 4 protons are pumped across the inner mitochondrial membrane to the intermembrane space from the matrix. Okay? putting all of those reactions together, then we see the mitochondrial electron transport chain in this diagram on the next slide. So slide 19, I believe it is. And in summary, we can say that electrons move from NADH or FADH2. So NADH if we're talking at complex 1, FADH2 if we're talking about complex 2, the cytochrome, complex 2, so succinate dehydrogenase, that in those electrons end up in the UQ pool, which is in the inner mitochondrial membrane, and then the UQ pool in the reduced form of ubiquinol transfers electrons to cytochrome BC1, so that cytochrome complex or complex 3, and then cytochrome C is ultimately reduced on the inner mitochondrial membrane space, and it can then transfer electrons to cytochrome oxidase complex 4, and that's where we see the reduction of oxygen to water. And then again, looking at the proton movement, we see that for every 2 electrons transferred to oxygen from NADH, 4 protons are moved at complex 1, and then also at complex 3, and then 2 protons are moved across the inner mitochondrial membrane at complex 4. Okay. So we can put that into an equation where we have NADH and oxygen, and then we see NADH being oxidized to NAD+, and then water being reduced to oxygen. That means that for every mole of NADH that's oxidized, there is energy released, so about 220 kilojoules, and we see less energy being generated, released, if we start with succinate being oxidized. Okay? That energy is used to pump protons from the matrix of the mitochondria to the intermembrane space, so about 10 protons are transferred for every pair of electrons if we're starting at NADH, fewer if we're starting at succinate. And those protons that are translocated set up a pH gradient and a charge difference across the inner mitochondrial membrane. And so it's important to remember that it's both a pH gradient and a concentration gradient with the charge difference, and that is a proton motive force. And that's what's used to generate ATP synthesis, and we'll talk about that in the next lecture. What I'd like to now do is return to this idea of structure and function. Now you are you've got those components of the electron transport chain back in your mind, okay? So now we can talk about their location in a bit more detail. Yes, they are on the inner mitochondrial membrane, but remember, the inner mitochondrial membrane has 2 subdomains. It has the inner boundary membrane, and it's got the Christie membrane. So a group of investigators looked at where the different complexes of the electron transport chain of the mitochondria are located, and where, for example, other proteins in the mitochondria are located. For example, the proteins that are involved in moving both nuclear encoded proteins and mitochondrial encoded proteins, okay, around the mitochondria. So remember that yes, mitochondria have DNA, but some mitochondrial proteins are encoded by the nuclear DNA. And those nuclear encoded mitochondrial proteins have to move from the cytoplasm, where they're translated from the messenger RNAs, right, into the mitochondria where they are used. Okay? So those proteins involved in the transfer are known as the TOM and TIM complexes. So TOM for the outer membrane, TOM, so outer membrane, the OM, and T for translocon, and the TIM, the translocon of the inner mitochondrial membrane. Right? So the group of investigators used immunocytochemistry, so they chemically fixed mitochondria, made thin slices of the mitochondria, and then labeled the mitochondria with antibodies against these different proteins that they were interested in localizing. And then they used a secondary antibody that bound to the first that had a gold grain associated with the secondary antibody. So in electron micrographs, they could see the gold grain as a black dot. So what's shown on this slide is an artist's representation of the labeling, so where each of the dots represents where the proteins are located. So here we've got the inner mitochondrial membrane with the inner boundary membrane and then the Christie membrane projecting out into the mitochondrial matrix. And so these investigators found that the proteins of oxidative phosphorylation are enriched in the Christie membranes. And so an example of this is COX-2, which is one of the subunits of the cytochrome oxidase, and that's shown in the right-hand side of the top left figure. So the artist's representation of the gold grains concentrated around the Christie membranes. And they quantified the gold grains, and that's what's shown in graph A on the right-hand side of the slide. So you can see the number of gold particles that they counted on the different types of inner mitochondrial membranes versus the Christie membranes. Okay. And also the outer membrane they included in their work. Okay. They found, on the other hand, that proteins involved in the insertion of nuclear and mitochondrial encoded proteins in the inner mitochondrial membrane. So that's shown in graph B on the right-hand side of the slide, except for a mitochondrial ribosomal protein and also a chaperone protein. So those are the last two proteins in graph B, where we see more of those in the Christie membrane. But all of those other proteins, and I've defined what the abbreviations are at the bottom of the slide, all of the other proteins were highly represented in the outer membrane and the inner boundary membrane. The investigators also found that the distribution of the proteins actually changes depending on the physiological condition of the cell, the physiological state of the cell. So they found that there was a high representation of TIM23, so one of the proteins of the translocon of the inner membrane, when proteins were being translocated. Okay? But when they stopped protein translocated, so when they treated yeast cells, so this was all done with yeast, when they treated the yeast cells to stop protein translocation, they saw that the TIM23, more of it, was found at the Christie membranes. So the location of the mitochondrial proteins actually changes depending on what the mitochondria are doing at any given time. Okay? So again, thinking about that connection between structure and function. Okay, in the last part of the lecture, I'd like to now look at the differences that we see with plant mitochondria, because there are some striking differences with plant mitochondria compared to other eukaryotic mitochondria. So the standard mitochondrial electron transport chain is found in plant mitochondria. So in other words, what's known as the cytochrome pathway that we've just been talking about is found in plant mitochondria. But plant mitochondria also have an alternative pathway of electron flow through the mitochondrial inner membrane. And that additional pathway, that alternative pathway, involves matrix-oriented NADH dehydrogenases and NADPH dehydrogenases. It also involves internal, or did I say? Okay, I'm sorry, I can't remember which one I just talked about. It has matrix-oriented NADH dehydrogenases and NADPH dehydrogenases, okay, as well as external NADP dehydrogenases. Okay, so these alternative dehydrogenases in both the matrix of the mitochondria as well as the intermembrane space of the mitochondria. Okay? In addition, plant mitochondria also have an alternative oxidase. Okay, so they have cytochrome oxidase, but they also have an alternative oxidase. The roles of these alternative enzymes have not all been completely worked out. We know what some of them do, but not all of them. So with respect to the matrix-oriented NADH dehydrogenases, they're insensitive to rotenone. So rotenone is a pesticide or an insecticide, and basically it stops the activity of complex 1. Okay? So these alternative matrix-oriented NADH dehydrogenases are not sensitive to rotenone. They keep on functioning, okay, even when rotenone is around, and that's how they were discovered. Okay? Electron flow was still happening in plant mitochondria, even though rotenone was being used to turn off NADH dehydrogenase activity. Okay? They have a very simple structure, much more simple than complex 1, it's much smaller. They transfer electrons like complex 1 into the UQ pool, but no protons are translocated across the inner mitochondrial membrane at the NADH dehydrogenases on the matrix side of the membrane. The affinity for NADH is lower than that of complex 1, so these NADH dehydrogenases are active when NADH levels are high in the matrix. For the matrix-oriented NADPH dehydrogenases, they're found in only some plant mitochondria. Not all species have these dehydrogenases, these alternative dehydrogenases. Again, they're insensitive to rotenone. They're much more simple in structure compared to complex 1. And again, they transfer electrons to the UQ pool. Okay? Again, no protons are translocated across the inner mitochondrial membrane. So, and in this case, no clear role has been determined for these matrix-oriented NADPH dehydrogenases. All right, what about the external NADPH dehydrogenases? In both cases, okay, so for both the NADH external dehydrogenases and NADPH external dehydrogenases, they're insensitive to rotenone, simpler complexes than complex 1, much smaller. They transfer electrons to the UQ pool, as shown in the diagram, but again, no protons are moved across the inner mitochondrial membrane. They appear to be co-expressed with specific alternative oxidase proteins, which we'll talk about in the next slide. So it looks like they might function hand in hand with the alternative oxidase. And they're thought to oxidize cytosolic NADH, NADPH, and in that way influence the cytosolic ratios of the oxidized and reduced forms of NADH, NADPH, and also the reactions that require NAD or NADH, NADP, or NADPH. Okay, so they have some kind of potentially regulatory function. All right, and then besides the dehydrogenases, as I've mentioned, there's the alternative oxidase. The alternative oxidase is actually found in all kingdoms except archaebacteria, but it's been lost from some vertebrates and some arthropods. Okay? It's insensitive to the inhibitors of cytochrome C oxidase, and again, that's how it was discovered. Okay? So when cytochrome C oxidase was inhibited by cyanide, for example, there was still mitochondrial electron transport going on. So besides cyanide, azide also inhibits cytochrome C oxidase, but not the alternative oxidase. Carbon monoxide and nitrous oxide also inhibit cytochrome oxidase, but not the alternative oxidase. It diverts, this alternative oxidase diverts electrons from complex 4, from cytochrome oxidase, and that's illustrated in the diagram. So electrons come out of the UQ pool at the alternative oxidase. They don't all go through complex 3 and complex 4. Okay? So electrons are transferred to the alternative oxidase, generating water. Okay, so oxygen is still the electron acceptor, but there's no protons being pumped across the inner mitochondrial membrane, and cytochrome CB1 complex is missed, as is the cytochrome oxidase. Okay? The energy from those electron transfers through the alternative oxidase is lost as heat. Okay? ATP is not generated because the proton gradient isn't set up through the alternative dehydrogenases or the alternative oxidase. Okay? So instead, energy from the electron transfers is lost as heat. So an example of this heating from the alternative oxidase, taking up electrons and passing those electrons to oxygen, is found in the thermogenesis that happens during the flowering of some plants. Okay? So here's a clear role of the alternative oxidase. If you have skunk cabbage, which is shown on the right-hand cartoon, or an arum lily or a voodoo lily, so arum lily on the left-hand side of the slide, the voodoo lily in the middle, all of these lilies have a part of the flower that's known as the appendix. And so in the diagram of the skunk cabbage, there's this very tall appendix. You can also make it out in the voodoo lily, and then the yellow appendix that's shown in the arum lily. Okay? There are lots and lots of mitochondria in the outer layers of the appendix in all three of these types of flowers and all three of these species. And those mitochondria respire through the alternative oxidase at very high rates when the pollen is being released by the male flowers, okay, of these species. This mitochondrial activity through the alternative oxidase heats up the tissue. So the tissues can be 10 to 25 degrees higher than the surrounding air temperature. Okay? And that heating up of the tissues leads to the release of odors from the flowers that then attract the insects that are the pollinators of the flowers. Okay? So the alternative oxidase activity in the mitochondria of these male flowers, okay, of these plants, allow the flowers to be pollinated. Okay? That means that seeds will set, and then that means, of course, that these plants can reproduce. Okay? So a very clear role of the alternative oxidase. And we'll stop there.